Hi, my name is Daniel, and I'm here to tell you about a new technique in biomedical research called single-cell RNA sequencing, a great example of the amazing power of OMICS. Let's start with the basics. Our genes contain all the necessary instructions to make cells and whole animals. Genes are transcribed, or copied into RNAs that leave the nucleus to be read by ribosomes to produce proteins that perform cellular functions. However, it's important to remember that not all genes in the cell are being expressed at the same time. To conserve energy, cells shift their focus from one area of necessity to another in the presence of varying stresses to achieve a multitude of diversified processes when the body needs the most. For example, a cell that's infected by bacteria needs to express genes implicated in defense. But, while in the presence of abundant food, it needs to express genes that allow them to store energy in the form of fats. By collecting all the RNAs in a cell and reading or sequencing them, we can identify the specific genes that are only expressed when the cell is being attacked by bacteria or when it's in the presence of abundant food. This technique is called RNA sequencing or transcriptomics. Omics meaning global because we're analyzing all the transcriptional events in the cell. This technique is also very useful in understanding development. Think of zebrafish, an amazing animal model that develops most of its organs in just 48 hours. By performing RNA sequencing at different developmental stages, we can determine the order in which genes must be expressed to make an animal. The problem is, even when we collect all the RNAs, we still don't know which ones come from cells in the brain, the liver, or the heart. We're lacking crucial information. To solve this problem, we have single-cell RNA sequencing. In this technique, each cell is trapped in a bubble that contains a specific barcode or tag. When cells are broken, all the RNAs coming from a particular cell are labeled with the same barcode. So now, even when all the mRNAs are mixed together, we can easily tell which ones are coming from the same cell. Now, we can create plots in which each dot represents an individual cell, and when we group the cells based on the similarity of their transcriptomes, we find that all the cells coming from the same tissue tend to cluster together. You can do these analyses at different developmental stages, in response to different types of stress or even under pathological conditions. So, this technique can give you critical information about gene function and disease. Well, my time is up, but don't forget about the amazing power of omics.